Welcome back to the studio. It's just 15 minutes till the tech update. We'll be seeing Rosemary Sinclair-Dokos and Lakmal Jayasinghe take the stage to update everyone in the community online and here in the audience about what nanopore sequencing is today and what we hope it's going to be in the future. So we're excited about that, but in the run-up to that, I've got a wonderful studio packed full of direct RNA action. We're joined by Mark Akeson, who is a godfather of direct RNA sequencing, and we're going to come back to you, Mark, at the end to celebrate some of the work that these guys have been doing in direct RNA. So we've got Chloe, Eva, Maria and Anna. Thank you so much for joining us. Firstly, can I just ask each of you what it is that you're doing with direct RNA? Chloe.
So I'm from the Pasteur Institute in Paris, and so we're interested in everything around bacteria and also bacterial resistance. So we use direct RNA sequencing to uncover gene resistance mechanism of action that we couldn't detect before. So it's a 15-year-old project that we brought back to life because the technology now allows us to sequence native RNA and get the modifications. So with this, we were able to uncover a new mechanism of action of resistant genes acting on ribosomes. So our theory is that this modification on the ribosome is likely to disrupt antibiotic binding and thus confers resistance to the strain.
And is this an unsolved problem in drug resistance?
For this particular strain, yes, because 15 years ago, this bacterial strain was detected and resistant to this antibiotic, but back then the traditional method failed to uncover why, how was this gene acting. We suspected that it was due to a modification on the ribosome, but we couldn't prove it. So now, thanks to direct RNA sequencing, we were able to detect it, which was not possible before. So, yeah,
fantastic.
It opened really new doors.
Well, this connects all with Gordon's opening talk when he was talking about the O'Neill report, which said that by 2050 we think that drug resistance is going to kill more people than cancer. Such important work, so thank you for doing it. Eva Maria, tell us what you're doing with that direct RNA sequencing.
Hi, so what we're interested is in understanding what is the role that RNA modifications play in the fate and function of RNA molecules, both at the molecular level, but also more at the level of like during development, as well as even across generations, whether they play a function in the transmission of information across generations.
Fantastic. And Anna, what are you up to?
So we're using direct RNA sequencing in clinics. So we hope to use direct RNA sequencing in clinics, and we could already use it to validate genetic cause by validating loss of function or loss of methylation in the ribosomal RNA based on blood samples. And yeah, we're investigating the performance of the direct RNA sequencing flow cells for using it in clinical contexts.
Fantastic. So you're all thinking about modifications in RNA, something that can't be seen unless you are doing direct RNA sequencing. So how does it feel? What are the good things and maybe the bad things about working on a completely new technology, a completely new method? Do you come up against stumbling blocks in doing that? Anna, has that ever caused you any problems doing this pioneering work?
So when starting with the 002 flow cell, I remember I was super amazed that we can see the base calling errors for the RNA modifications, which was super cool. But then further going to the 004 flow cell, we couldn't see these base calling errors anymore, not all of them, but it was a challenge because of the fast development of the tools or the techniques from Oxford Nanopore technology to really use it or develop methodologies or for analysis. The main challenges we had is validating the RNA modification detection tools that are available or choosing the best one. I mean, now there are RNA modification detection tools by ONT itself, but there are others that might perform better, might perform worse. I think it's a challenge to find out which is the best model to detect RNA modifications in the context of interest.
I think what you're saying really underlines the importance of Oxford Nanopore being able to work with the community, because I think that the work that you'll see today announced in the Tech Talk is as a result of listening to people working their technology and us trying to rapidly improve it and get it better and better and better so that we can solve these problems and you guys can put out your nature papers that we all love to see. So Eva Maria, you've been working a lot on method development and sort of validating these data sets. What do you think the opportunity is then for working on a completely new method for you in your position in science? So what's the opportunity for working on a completely new method with the programs that you're working on?
So, I mean, I think that right now having the possibility to do much more throughput really gives the possibility to start asking questions that before could not be answered. Because if you want to do studies at the transcriptome-wide level, for example, in human samples, you could only look into very highly expressed genes due to the coverage. And I think that a major achievement with the new chemistry is really the increased yield, and also the flow cells are much more stable, so you can even reuse them several times. So this really makes it possible to start answering questions about what is the function and role and distribution of modifications that before could not be done easily, or you would have had to spend a lot of money in doing many in putting many samples and many flow cells.
Fantastic. And Chloe, how are you feeling like pioneering a completely new piece of science within the microbiology world?
It's exciting.
Yeah. Do you have to work hard to explain to people what you're doing?
Depends to who, but yeah, I feel also like it's still a bit niche also to apply direct RNA sequencing to bacteria and viruses. But there's, I think, a lot of application in this domain, in this field. For example, now we have the ability to sequence RNA viruses, which is something we couldn't imagine before. We had to go through reverse transcription, amplification, amplicons. Now we can directly sequence viruses.
Fantastic.
So, yeah, super exciting.
Mark, I have to ask you, you've been working on direct RNA for such a long time as a pioneer and a developer of the method, and I know a user of the method as well. How does it make you feel to listen to some of the work that's happening out there in the community?
It's incredibly gratifying to see talented people pick up on this technology, and I can't overstate that. I can give you 2 reference points for the technology that kind of bookend this. The first experiments we did with Nanopore in 1996, I was involved when used RNA. It was poly-A-RNA and poly-C-RNA, and it took us a year to get one experiment to work. And that experiment was recorded on an oscilloscope, and not recorded, actually, it was observed on an oscilloscope, and we had no way to record it. And so it took us a year. We saw that there was a difference between the 2 classes of RNA, but it was not stored in any fashion. And then you wind this way ahead till now, and to give you a reference on how powerful this technology is, we have a colleague named Harry Noller, who is one of the people who's characterized the ribosome and spent 30 years at it, and basically revered in that field. And he came up, in front of a group of people at our RNA center, was talking about RNA being sequenced with a nanopore, and he said you can look at 1 molecule at a time. And he was absolutely astonished by that. So that's a reference point. And now what I think this kind of work is going to allow is people make discoveries that are totally unanticipated. And so I think that's just an amazing thing, and it's really a credit to the people who are picking up on it now.
Fantastic. Well, we are nearing the Tech Talk, so I have to ask you, what is it that you would like to see announced in the Tech Talk that will improve and enhance and scale your direct RNA-based research, Chloe?
The ability of multiplexing different samples on the flow cells. Definitely that would be something very useful for us, especially because we work on small organisms, small genomes, so being able to have many samples on the same flow cell would be I think really needed.
What would you do with it? Would you do more of the same bacteria, or would you spread your wings into broader pathogen or microbiology samples?
Yeah, definitely spread it. I think, to different viruses, different bacteria, and I think that would make it much more accessible if we could multiplex samples.
Fantastic. What about you, Eva Maria?
I would like to know which additional base calling models they plan to release and when. Mm-hmm. I've heard some rumors, but it would be nice to know the ones that are maybe potentially going to be released today, as well as maybe the future ones that they plan to release maybe for the future London calling.
Fantastic. Anna, what would you like to see in the tech talk today?
I think I would have said the same answer, looking forward to maybe more RNA modification models for direct RNA sequencing, or I think that's it.
Fantastic.
Hopefully there will come more, but let's see.
And Mark, what would you like to see in the tech talk today, or indeed the tech talk in 2 or 3 years' time for direct RNA?
I would like to see how far advanced the machine learning has gone to get better accuracy and to incorporate modifications of all sorts, including what are called the 2'-O-methyl modifications. And that really adds a whole new level to what Nanopore can do.
Fantastic. Well, you saw from Gordon's opening talk that we are really deploying machine learning as much as we can to accelerate the analysis of these data sets and to obviously create new algorithms that can get as much as possible out of that amazing raw signal that we get out of our little nanopores. So hopefully we can give you all of your Christmas presents today. And thank you so much for joining us on the sofa today. I hope you have a good tech talk, and I hope you have a good conference day tomorrow as well. Thank you very much. So thanks everyone online for joining us today. We're going to wind down here now in the studio and pass over to the guys on the stage presenting today, and we'll join you here in about an hour and a bit for a review of the tech talk and a conversation about CNS tumours and how we get that towards everyday clinical practice. Thank you very much.